neurodegeneration. And so we'll proceed by getting a handle on what we currently have in our portfolio, which is what Rosa will cover this morning. And then this month, we'll hear from ALS expert Jeff Rothstein and Parkinson expert Lorenz Studer, both pathfinders and researchers at the cutting edge of their field, scientifically and medically. And then next month, we'll hear, I hope, from Alison Goat on Alzheimer's disease, and Sarah Tabrizi on polyglutamine diseases, particular Huntington. And we'll have some time to begin discussing what our next steps might be in this area. So without further ado, let me give you Rosa. Rosa, you've got about 25 minutes to work on the neurodegenerative review, please.
Okay, thank you, Larry. And Kelly, can you share the slides, please? We have a tandem situation so that I could access my screen. Thank you, Kelly. Fantastic. So thank you, Dr. Goldstein, members of the Neuro Task Force, Dr. Studer as well. It's an honor to have you with us and the public. I will be providing a comprehensive overview of our neurodegeneration portfolio analysis that has been developed through concerted efforts from Dr. Crisis, myself, and our dedicated program teams. Before we dive into the specifics, I actually would like to take a moment to acknowledge the work of our colleagues, Dr. Janie Bynum, Dr. Kelly Shepard, Dr. Sarah Taylor, Dr. Lisa McGinley, for their meticulous work on putting all of this together over the past months, and for their compilation of the analysis. And I would also like to give a special thanks to Dr. Shyam Patel for providing the partnering data in the last slide, which helps us emphasize the impact of some of our work. So without further ado, let's get this started. Next slide. This is one of the 3 slides, and I'm going to focus on this one. The 2 others are basically, this one represents Proposition 14 and Proposition 71 numbers, and the next one is just Prop 14 one, and the other is Prop 71. So I'll be going quick through those, especially as we have only 25 minutes. We have, so this slide represents the scope of SEM's investment in neurodegeneration, combining the efforts of both propositions with the years from 2007 to 2023. We have divided this slide into 3 main columns: the diseases addressed, the number of awards given on the left, and the funds allocated as denoted in millions of dollars on the right. The color coding, which is going to be consistent across the presentation, for better visualization, shows discovery awards in yellow-green, translation in orange, and clinical awards are in blue. So to the left, with the number of awards, what that highlights is that Parkinson's disease has received a total of 28 awards, with discovery being the focus. Alzheimer's follows with 17 awards, also heavily focused on discovery and some translation, not clinical. ALS has a notable number of 29 awards, again, with a focus on discovery, but also with a significant portion in clinical trials. And then we have Huntington's and spinal muscular atrophy, which have received a combined number of 23 and 4 awards, respectively. Looking at the number of funds, ALS has the highest investment with over $106 million, emphasizing the focus on both equally, more or less, discovery and clinical trials. Parkinson's has also received a considerable amount of funding with a total of $63.9 million, where 44.6 is directed toward discovery research and 15.3 to clinical. Alzheimer's disease has $50.2 million in funding with a balanced distribution between discovery and translational research. And Huntington's disease has been allocated $49.1 million, again, with a strong clinical trial investment. The lower section of the slide identifies a focus on rare diseases, such as Tay-Sachs, PML, and lysosomal storage diseases, And most of them have 1 to 2 awards each and corresponding funding. And this indicates that while these conditions are less common, they are also a focus of CERM's efforts, as we all know. Some of the investment on neurodegenerative diseases, as you can see, is in the discovery rather than clinical. For example, Alzheimer's disease versus ALS, and this is due to some of the factors, but Alzheimer's disease compared to ALS has seen more of a significant advances in cell and gene therapy approaches, given its clearer genetic components in some of the cases, which has allowed more targeted therapies. And we will hear about these today from Dr. Rothstein later on, I'm sure. But in essence, this slide provides a comprehensive overview of our strategic funding distribution across a range of neurodegenerative diseases across a 16-year period, with significant investments in both early stage and clinical research, which highlights our commitment to neurodegenerative diseases. Next slide, Kelly, thank you. So this slide represents the number 2 and 3, as I said earlier on, is the CERN neurodegeneration portfolio of awards data under Prop 71 in this case. And the main observation is that the underlying trends in funding and award distribution remain consistent across both timeframes, and with the previous, that was the overall picture between both propositions. So let's go past again, the next slide. This is Prop 14, and again, showing that the underlying trends in funding and award distribution remain consistent as well right now. Given that we are only a couple of years on this proposition, we have less a number of awards and less amount of funding invested. Next slide. On this slide, we are looking at the CERN neurodegeneration portfolio spending by disease, comparing propositions, contrasting the allocations on the Prop 71 on the left and Prop 14 on the right. The slide highlights the distribution of funds across the spectrum of neurodegenerative diseases, with a focus on how financial resources have been directed towards either discovery, translation, and clinical research. And again, is yellow, green, discovery, orange, translation, and blue is clinical. So on the left, on the proposition 71, you can see the Parkinson's disease, disease received the highest funding in discovery, with a significant portion channeled into clinical research as well, demonstrating CERN's commitment to translating also the discoveries into potential treatments. Alzheimer's disease also saw a substantial investment, especially in discovery and translational research, which reflects the strategic emphasis on unraveling the complexities of the disease. And ALS, it has a nearly equal distribution of funds across discovery and clinical stages, which received, and ALS received the highest amount of funding. The equal distribution underscores the balanced approach towards developing, as well as understanding the disease mechanisms and pushing the promising treatments into the clinical. And the significant clinical investment is likely a response of the urgent need to find therapeutic options for this rapidly progressing condition, I would say. So moving on to proposition 14, the funding pattern here shows the continued focused boost in clinical research for ALS. And to note, we are comparing 17 years versus 2 years, so Prop 71 versus Prop 14. So we cannot really make any conclusions yet. Next slide, Kelly. And feel free, if anybody has a question or wants to clarify, please stop me. We are going to move into funding. The next 3 slides are going to be about Prop 71 and Prop 14 together. So this slide displays CERN's R&D funding in neurodegenerative portfolio as a percentage of the total in each pillar under both propositions over the last 17 years. And it showcases the allocation across discovery, So 43% of the funding of Neuro for discovery has been in neurodegeneration. 26% has been for translational, and 23% of the full Neuro funding in clinical has been in neurodegeneration. Discovery shows the largest share of the neurodegeneration specific funding at $169.8 million, which emphasizes the importance of foundational research. And I could go further and say this is discovery of new disease mechanisms that are very needed for all these diseases, complex diseases. And translational efforts have received the 61.8 million, which bridges the lab findings into the clinical applications. While clinical research is the most resource-intensive phase, and it has allocated 96.2 million, reflecting the significant cost of bridging therapies to patients. Let's move to the next slide. The next slide shifts our focus from funding to the distribution of awards within CERM's R&D neurodegeneration portfolio from the duration of the last propositions of 17 years. It outlines the numbers of awards given in the 3 categories again, and versus what we have spent in total neuro. You can see that in the discovery category, there have been 90 awards specifically targeting neurodegeneration, which combined with other neurological awards totals 218 awards. This highlights CERM's strong emphasis on foundational research in neurodegeneration. Translational research has seen 11 awards dedicated to neurodegeneration out of 37 total neuro awards, and reflects the focused efforts to bridge the gap between laboratory findings and potential clinical applications. But it also shows that many of the discovery might not be successful, no, because we are not translating them. Or they might be being the risk somewhere else in for-profit or industry. In the clinical stage, there are 10 neurodegeneration specific awards from a total of 41, and this shows the strategic push towards bringing the therapies from the lab into the clinical testing phase. And this award distribution, I think, complements the funding patterns that we discussed earlier, demonstrating CERM's balanced investment in each research stage to advance the understanding of these devastating diseases. Let's move to the next slide. The next slide is an alternative way to visualize the number 5, and is, instead of percentages, is by amount spent relative to each other. So it's all the same as the first slide on this trend. Next slide. This slide provides an analysis of the percentages of neurodegeneration spending compared to the other neurological areas in the most recent funding phase under Prop 14. So the last 3 slides were both propositions. This is just so we can see what's happening with Prop 14. And here we observe that the strategic allocation of funds parallels the trends of both props together. Next slide. This is in number of awards, and the distribution also parallels the trends that we had under Prop 71 and Prop 14 together. Next slide. And this is by spending another alternative way to visualize slide 8, instead of percentages, by amount of spend relative to each other. Now, any questions so far? Okay. So if there are no questions.
Yeah, actually, one question, Rosa. What fraction or approximate fraction of the CLIN awards are partnered with industry?
We will see that at the end. We're going to see that at the end. So we're going to see a couple of things. Thank you, Larry, for the question. So one of them, we are going to see the progression. So those awards that have started with funding from CERM at the stage of either discovery or translational, and they've moved to the next stage, also funded by CERM. And another set of data is going to be about partnering activities. And that's what Dr. Patel provided, and we will discuss this. So let's move on to the next slide. This is a busy slide.
Rosa,
Yeah,
this is Keith.
thank you.
I'm sorry to be late, so you may have already talked about this, but what fraction of I assume that the clinical awards include trials?
Correct. Yes.
And so what fraction of the expenditure on the clin side is for specifically for supporting clinical trials?
So we could be, let me see, on the clinical, Abla, can you move to, Kelly, can you move to slide number 5? And Abla, I will defer to you because I would say that it's 23%. No, number 5, slide number 5, Kelly. This one. Abla, would you like to answer the question? I would say it's 23%, but I don't know if Abla, you might be muted, Abla.
I'm no longer muted. Yes, as represented on that slide, it's 23%. In clinical trials, that's his.
Yeah.
I think saying they're in clinical trials is just a little misleading because the clin awards include the studies leading up to an IND filing. So you have to have an IND in order to actually begin clinical trials. If you go to those slides before, you'll see that there are two $4 million amounts. Those are all studies leading to an IND. There's only one that's, so that slide, for example, the blue ones, the two blue fours have to be trials leading up to an IND. So whether they get the IND or not is still an open issue. So then there's one for 12 million, which suggests, unless there's more than one involved there, that's probably a CLIN award, probably a phase 1.
Yes, you are correct, Steve. And
Yeah, very helpful.
I think we did that analysis. I would need to go into other slides, but we could follow up with providing this information.
Okay.
But I don't think somebody is speaking. Sorry.
Yeah, I think the focus of this task force is on Prop 14. So are you going to be focusing what we've done so far related to Prop 14, or are we just looking at a combination and then we have to parse out how much is Prop 14 and how much is Prop 71, which is not really part of the scope of what our task force is charged with dealing with. Just wondering how we're going to get to that when all of these numbers are combined.
Thank you, Fred, for your comment. Actually, the numbers are separated. We've done the 3 analyses. We've done combined and one by one, so that we could see what has happened so far, what happened during Prop 71, and what's happening during Prop 14, so we can see whether the trends are still the same. And for now, we can see what we are concluding is that the trends are the same during Prop 14.
Okay, thank you so much.
Yeah, of course. And I think that's a very relevant question. So you're absolutely on the spot. The thing is, this is a lot of information. That's why it was posted, so that people could digest it before the meeting, because going through all these slides is kind of a little bit too much information, right? So I am going to go, we were going to go to slide number 11, where we are presenting. So again, the question that was asked in terms of the proportion by Keith Yamamoto and Steve Julesgaard, in terms of clinical, we can say that we have funded 23% of the whole Neuro portfolio, with a correspondence of $96.2 million have been invested in clinical. And of those, about half has been in ALS. So we can, that's all what we can say. And then we will have, in terms of ALS, for example, we can see here that in terms of number of awards, we have 3 in the clinical. This slide is actually showing by approaches. Is probably, and it's a comprehensive overview of the neurodegeneration portfolio of awards by approach on the Prop 71. We also have Prop 14, Fred. And we have categorized these in 4 columns representing disease on the left, and then the 3 stages: discovery, translational, and clinical. And it breaks down, the chart breaks down the number of awards given to various neurodegenerative diseases. For example, we can see that for Parkinson's, we received Parkinson's disease, they had I think 23 awards in the discovery phase, 1 in the translational phase, and then we have 2 in the clinical phase. And then we have the color-coded legend at the bottom shows the different research modalities, such as allogeneic, autologous, gene-modified, small molecules, cell-free gene therapy, foundational research, and autologous biologic. And these modalities will help us understand where the focus of our research also has been. I would like to draw attention to ALS. It has 13 awards in discovery phase, which are highlighted in, not 13, 17 awards in the discovery phase. And we also have 3 in the clinical, but given that it has, so those are probably clinical trials because of the number of awards and the amount that we've spent in ALS clinical.
Can I comment here on when it comes to the clinical, it's actually just phase 1. And so it is like uncontrolled clinical research in ALS. Defining there are no placebos, whatever. So if whenever you call a trial, I think it's fair to say that you need a comparator. And in our situation with ALS, other than Brainstorm Therapeutics, the other 2 were only research, open label trials, open label research.
Great. Thank you for the clarification, Abla. So the next slide actually shows, instead of number of awards, it represents the portfolio by funding, by approach, and this is a stage, and again, also the different color-coded modalities. And as you can see, this kind of parallels the amount of funding that you were talking about in clinical. We can see that we've spent most of our funding in clinical, in Parkinson's, ALS, and Huntington's disease in Prop 71. Next slide. This slide, Fred, is the same, but just for Prop 14. So for Prop 14, we can see that so far we have had quite a bit of investment in clinical as well. So Parkinson's disease, we have 1 award in clinical, we have 1 in ALS, and we have 1 in Tay-Sachs. We have some translational approaches for Parkinson's again and Friedreich's ataxia. And then we have in discovery 1 award for Alzheimer's, multiple sclerosis, a lot of funding or a lot of awards in ALS. So it's like it's one of the candidate neurodegenerative diseases that we have invested the most. It's actually the one that we invested the most, ALSP and PML. And you can see the different approaches with the code at the bottom. The next slide shows the same information, but by funds spent. And we can again see that ALS is the one that has received most funding. And it's focused on early research, foundational or small molecule, biologics, and gene therapy, cell-free gene therapy. So they are these two awards trying to develop new candidates that will move into translation and clinical, not so much foundational discovery. And in the clinical, we have 12 million, and I think it's allogeneic gene modified therapy. And I think we will see an example soon in the slides. Any questions?
Rosa, you're getting tight on time.
Okay, so I'll just go. Thank you for next slide, Kelly. This slide provides an overview. I'm not going to go through this one. I'm going to go through the next, but what I want to present is what this slide and the next represent. These are the overall neuro portfolio progression of events for both propositions. What a progression event is a measure of the success of our programs and funding moving to the next stages. Now, what we capture here is what moves to the next stage within our CERN portfolio of funding. So what we haven't captured here, which we will try to capture next, is what might be the risk that goes then and is funded through industry, right? So a progression event is when a CERN award completes one stage of research and is successful and gets CERN funding to move on to the next stage, to develop either a second generation or of the same device or therapy. So in here, I would like to highlight a very a case study to demonstrate how our funding, when applied to programs such as the one pioneered by UCSF under Dr. Kriegstein and Dr. Rubinstein, that moved to Dr. Cory Nicholas and Neurona Therapeutics. They develop through 5 basic and discovery awards, they developed the protocols for derivation, characterization, selection, and production of interneuro precursors. Then they got the TRAN award, where they developed the inhibitory neurons as a therapeutic for focal epilepsy. And then they've moved on to phase 1/2 trial to test safety and the efficacy of the potential treatment for drug-resistant unilateral medial temporal lobe epilepsy. So this is a very nice example of how successful some of our neurotherapies have been evolving through our portfolio. Next slide is a focus on our neurodegeneration. And I will say we had made a mistake that we rectified. We had missed a couple and we added them. So that was what the revision of the slides was. And we can see here that we have a fair amount of Parkinson's disease. We have 3 projects that have moved from discovery either to TRAN or to CLIN, or that they are moving from discovery foundational to development candidate and so on. So this is just for reference. I just want to move to the last slide because I'm short on time. And this last slide provides a comprehensive overview of the neurodegeneration partnering activities from 2007 to 2024. And by partnering, what we mean here is that a program that had started with CERN funding has either raised money through a VC, spun out of academia into a for-profit, and or raised VC funding or licensed or been acquired. And this is a measure of the impact of CERN funding. And these partnerships, I think, are vital for advancing research from the lab to the clinic, emphasizing the importance of collaboration between public institutions and private entities in tackling these devastating neurodegenerative diseases. And I think it was, I can't remember who asked the question, but you can see that, yes, in the clinical, we have some programs that started in the clinical and that have had partnering activities. So you can see in Huntington's, Parkinson's, ALS, Parkinson's disease, we have details, but we have some in each. Stephen.
Yeah, so some of these descriptions of where things stand are a little misleading. I did my own independent research on all of these companies. I want to start with Brainstorm because actually they completed a phase 3 clinical trial, but had an FDA advisory committee meeting which voted 17-1 not to proceed, for the FDA not to proceed with approval based on their phase 3 trial. So they're back to the drawing board. the Accurex, I'm sorry, no, let's do this. Aspen Neuroscience, they have an IND approved, but they're not in clinical trials yet, even though it says clinical trials ongoing. They're not there yet. There's another one that says that they're in clinical trials. So that's the Accurex, the second one down says clinical trials ongoing. That's not quite the case. They're trying to raise money right now to get into their first clinical trial. So anyway, my point is that I don't think we're nearly as far along with respect to industry partners as this slide might suggest. We're still way back at the very beginning of things, and the one that was the furthest along actually failed in terms of its phase 3 clinical trial. So we have a long ways to go in the clinical area, whether it's with academia or partners, before we're going to get anywhere in this entire area.
Can I answer, Stephen? Stephen, you're absolutely correct. Brainstorm, the phase 3 trial failed. They're still discussing, they're getting a special kind of management of a new trial with the FDA. So you're right, there are no clinical trials ongoing regarding that. I think that the clinical trials ongoing was for MS. Maybe the word MS was missed. The Aspen Neuroscience has applied for a CIRM grant and was approved. It's starting a clinical trial with us, and that's public. So just to correct those two. And so again, Aspen is again started their phase 1 clinical trial with CIRM funding.
Well, they've opened the phase 1 clinical trial OBLA, but they haven't recruited any patients yet. That's the issue there.
The design there's Steve. The design there is these are iPS cells where they already have identified the patients. They have already banked their iPSC cells, and they are going then to treat them so it's autologous. And that's what's going on. So the recruitment of the patients is part of the modality by which they're working with.
So given the time, I'm going to ask that Abla and Steve get on the same page between now and the next meeting, and we'll have time at the next meeting for further discussion of the portfolios. But unless somebody has a burning question, I'd like to move on to our presentation from Lorenz Studer, so we don't get too far behind on our schedule. So anything burning, guys? Okay, good. Rosa, thank you very much. That was a very comprehensive presentation, and I would encourage folks to work out some of the differences behind the scenes, and then we'll have another discussion of this, perhaps more in depth, at our next meeting in, let's see, what month are we in? March, so April. Okay, so our next presenter is Lorenz Studer, who I'm sure is known to most of the members of this group. Lorenz has worked on differentiation of dopaminergic cells and neurons for many years through all phases of his training. Of note, he's received a MacArthur Fellowship. He's a co-founder of Blue Rock Therapeutics and has done a great deal of work on the development of dopaminergic neurons for treatment of Parkinson's disease. I've also asked him at the end to cover some areas other than dopaminergic treatment for Parkinson's disease, where he thinks there are opportunities that are under-recognized and under-resourced for us to think about. So, Lorenz, I hope I got most of that right, and I'll turn the microphone and presentation over to you.
Great. Thanks so much for the invitation. I hope you can see my slides. Is that okay?
Perfect.
Okay, excellent. Yes, what I would like to do the next 30 minutes, if I have that correct, followed by 15 minutes of discussion, give you a little bit of an update on cell therapy development, focusing as an example, obviously, on our own work, but also broader status of the field. And as Larry mentioned, go a little bit beyond just dopamine neuron replacement towards the end. So just to get started, I have to put up my disclosure slides because we did actually spin out a company, which is Blue Rock Therapeutics, that was acquired by Bayer a couple of years ago, and that sponsors the work now moving forward, including the clinical work. And so this is, again, the broader outline of what I want to do. So really, we'll spend maybe about 10 minutes or 15 minutes really on kind of the core of where we stand with classic dopamine neuron cell replacement therapy, but then talk about some of the remaining challenges of that approach, possible next generation products, some of the limitations of the field like animal models, and then again go hopefully a little bit into the non-dopaminergic features that potentially could be treated by cell therapy approaches. I also want to state from the beginning that I really focus pretty much exclusively on cell-based approaches, and I'm not going to basically discuss disease modeling now using stem cells and so forth, which is an area we're also very much involved. Happy to discuss. But for today, I want to be focused on cell-based therapies. Just for an introduction, I'm not going to spend much time here. No, the rationale is pretty clear that what you want to do is you want to replace the dopamine neurons that are lost in the disease. And the reason why it's a widely discussed approach is because you actually have relatively few of those in a healthy person. You have about 500,000 roughly on each side of the brain. Once you lose half of them, you start getting some of those movement-related symptoms. And we all know it's a very common disease. It's a big socioeconomic burden on the society. Now, it's important to say, and that's listed here, that it's not a purely dopaminergic disease. And again, that's why I want to discuss maybe what we can do there. It's also then important how to state now what a dopamine neuron replacement therapy could achieve in Parkinson's disease. Even in the best dream scenario, now it would be basically a cure, quote, of the movement disorder, but it would not be a cure of Parkinson's disease, because again, Parkinson's has some of those other symptoms shown here that can often precede movement disorder-related symptoms by many years, actually. And some of them are obviously very feared. Happens not in all the patients, but quite a few of the patients actually develop major cognitive problems at later stages of the disease. So this field really started many, many years back as an idea, which is the cell replacement idea. And there was very important pioneering basic work done using, at that time, fetal dopamine neurons, work in the late 80s, 90s. And those studies are important because it kind of showed the paradigm that we really want to pursue. They showed that you can have fetal dopamine neurons isolated from fetal tissue. They show that those cells can survive in a Parkinson's brain. And these red blobs here is a tracer that labels the dopamine cells. And importantly, even though this patient for 23 years of the life after grafting did not receive any immune suppression, you can see a very nice surviving graft there at the very late stage of that patient's life. So the point that I'm trying to make, unlike many organ transplantation approaches, in the brain, you're probably going to get away with very little immunosuppression, and that makes it actually, again, also a good target. Now, many of you probably have heard of fetal tissue, and clearly it's not something that's routinely used in the clinic, and it has to some extent failed once it came to placebo-controlled trials. Happy to go into more detail if anyone is interested, but a big part of it is that the way the clinical trials were designed, they had the readout probably too early, because this is like, call it like a living drug. You know, the cells, they are alive, and they actually get their full functionality only about by 2 to 3 years after we inject those. So it's very, very slowly becoming fully functional. And some patients also developed side effects, and that really what's killed the approach, so-called graft-induced dyskinesia. So the combination of those questionable efficacy and potential side effects kind of stopped the field, but not completely, because neurologists followed those patients still longer time and found at least a subset of those seem to be doing very unusually well. Now, admittedly, these were very few patients, but there were some patients, for example, that could stop their dopamine therapy for many years. And that's something that's very rare in a disease that's constantly progressing. Now, we should soon hear from a study that was done by Roger Parker that actually went back and say, okay, did we learn from those early studies before 2000? can we select the patients better and then show that this was really the problem why the study failed, maybe different clinical trial design. And again, this is still unpublished data, so I cannot go into too much detail, but I don't think, again, that this study is going to resolve it, because I think even there is kind of unclear how good it really worked. It's still an uncontrolled study. And they had also a number of challenges that had to do with devices, with consistency of the function and so forth. So I don't think this is a resolved issue. And I think the main thing we learned, and Roger could speak more to that, is that it's really currently nearly impossible to do that with fetal tissue, because you apply similar standards of quality control to fetal tissue. It's very, very difficult to do that in a way to routinely treating patients. So that's then why we and others try to develop pluripotent-based approaches. That goes 10 years back, where we showed we could make those dopamine cells either in a way that they can rescue and survive in a mouse brain, in a rat brain, or in a monkey brain. They can do that in quite large numbers. They can restore some of the behavioral assays. They come back to a little bit what's the problem with some of those models. But there's one model where the mouse has kind of Parkinson's, you're going to call it, on one side of the brain. And then it spins around its own axis if you stimulate it. And you can use that as a readout. So if then the one side that has the disease gets grafted, it becomes symmetric. Or this is a rat where you see on the left paw that if you look on this cycle, it cannot really properly initiate movements in this paw. But after grafting, it can do that again. And finally, and again, I'm not going to spend too much time on that because it's a bit technical, but we also know exactly how these cells function. So it's not just some kind of a weird cell where we hope that it's positively influencing the brain. We can actually put the cells in with like a light switch, where the cells go into the brain, they rescue all those behaviors, but then we can literally flip the light switch and switch them off. And we showed that the animal is again fully Parkinsonian. So what those studies show is, in this case, it's really the nerve cells that integrate into the brain. They secrete dopamine, and they secrete it to the right cell. And so that's, again, one of the few diseases where we actually have a lot of this mechanism where we know exactly how these cells function. From there, it took us about another 10 years to really get that protocol that worked in those mouse, rat, and monkey in a way that we can use it in patients. So there's all these manufacturing issues that we resolved. We got an award from the New York State program, which is a $15 million award to really make a clinical grade product. We called it MSK-DA01. And as you probably all know, you have to do a lot of testing in animals. You do tumorigenicity testing, biodistribution, toxicology, hundreds of animals. So these are $1 million of studies. We do it in the rat, where we had something like 50 rats, where we had to show that we can rescue the behavior. And we did also a number of monkeys to show that the device we're going to use in the patient actually can reliably deliver the cells in a large brain, such as in a monkey. And I show you just one picture here, just because it's very easy to understand. So it's again this model where you have Parkinson's-like issues for movement on one side of the brain, so you lack this brown color, just the dopamine fibers. But once you graft our cells that were completely generated in a dish, human-derived cells, you can see not only the cells here, this darker area are the cells, but then they reconnect with the whole brain. And again, they do that effectively here in many, many male or female Parkinsonian animals. So that led us then in 2021 to really graft the first patient. In late 2020, we got approval from the FDA. That shows you just basically how that works. So they come from the GMP facility where they are prepared. They're brought in the surgery room. They are loaded into a device that we adjusted and optimized for delivery of those cells by Dr. Tabar. It's the neurosurgeon in this study and also did a lot of the preclinical work. You have to carefully check that the cells are properly delivered, and eventually you inject it directly into the brain. So you actually have to inject it into the brain parenchyma very, very carefully, obviously, to make sure you don't cause any bleeding and so forth. And you inject basically in each patient on each side of the brain 3 tracts. And each of those tracts has 3 deposits, so 9 deposits on each side, and 18 deposits then in total per patient. We did that in total of 12 patients, so it's something like more than 200 deposits were done in this clinical trial. And I'm happy to say that we didn't get any complication with regard to bleeding, hemorrhage, or anything like that. Now, this MSKD01 product has now this little bit unpronounceable name, bemdaneprocel. That's now the product name that Blue Rock takes it forward. And what I can tell you, and again, I'm not going to spend much time on that, these are some of the results from this phase 1 study that are not published, but they have been publicly released by the company, showing just that generally was safe and well tolerated in all 12 patients. 5 of those patients received a lower dose, 7 a slightly higher dose. We could show that this delivery can be done safely. I mentioned again, for example, no bleeding. We transiently immunosuppress the patient, but again, only transiently. And it was again very well tolerated. We have evidence that the cells survived. You remember this red color on the fetal grafting. So we have similar data for those, showing that they can basically increase this dopamine signal. And we have some suggestion. This is a phase 1 study, small groups, but you have suggestion that there might be actually an effect on clinical impact and maybe a greater effect in the patients that got more cells. I'll show you one slide after that to illustrate that. We didn't see the side effects that were seen in those fetal grafts in the past. And again, the caveat, this is still a small sample size. The primary goal was safety and feasibility. And in fact, in this regard, the study has already met, as you call, technically met its primary endpoint and can now move on to a later stage study. I'm going to show you 2 slides with regard to the early signs of potential efficacy. For the cohort A is the lower dose group, the cohort B the higher dose group. And this is one of those measures. So these are patients relatively severe. And even with the best treatment, they spend about 4 or 5 hours of the day so-called off. So they cannot properly do their activity of daily life, properly move around and so forth. So they are off. What you can see is that they got nearly an additional, we could reduce that 4 to 5 hours by about 2 hours in the higher dose. And this is kind of the converse one, so-called on time. Obviously, if they're not off, they're on, but they are on also without any of those dyskinesia that you sometimes see if you get on with a drug. What's also exciting is that trend seems to continue. So these were the data just released about a month or a few weeks, actually, less than a month ago, from the 18 months data. By now, it's actually 2.7 hours in the high dose, the off improved, and 2.7 hours in the on. And so that's really important because by 12 months, we removed the immunosuppression. So that suggests, again, if you believe this is a real meaningful effect, goes in the right direction, cells continue to mature, seems to have better effect, and the immunosuppression did not impact that, the loss of immunosuppression. That's another score. Again, many of you might not know exactly Parkinson's score, but it's the most widely used Parkinson's score. On a scale where these patients have typically about 40 to 50 points, the more points you have, the more severe you are. And you see that by 12 months, particularly in the high dose group, there seems to be a trend that this gets better by 13 points. And even the worst patient shown here was going a little bit down. And again, usually the disease progresses, you slowly increase this score. But what's really exciting is by 18 months, the high dose cohort went all the way to minus 23 points, which is quite a big point change. Again, we have to be very careful because it's a small group of patients, but it's at least as good as we could have expected from a phase 1 study. And so we're really excited to see how that moves forward. So the way it's supposed to move forward is that we're now moving to the next phase of study, which is supposed to start in Q3 of this year. And that will be a placebo-controlled study. And there's again some challenges how you actually do that for a surgical trial, putting cells into the brain and so forth. Happy to discuss that. But this will again start very, very soon on a large set of patients and many more centers than the early study. Now here we get then again how this compared to other ongoing efforts in the field. And I think you discussed a little bit what CERM already funded. So there has been a trial started in Japan, actually quite a long time ago, but they have a slightly different approach. So unlike our group where we make something like 10 billion cells off the shelf, then we just go back to the freezer and take cells for every patient. They had to prepare the cells for individual patients because they couldn't freeze the cells at the end point. So they haven't really reported their results yet, but they have dosed 7 patients, and I think they are at the 1 or 2 year mark. So we hopefully soon will hear from them. And they actually want to do a similar early stage, I think a phase 1 trial. They apply for the FDA doing that in the US. There's another group in Lund, and actually those three groups, together with our group, were the so-called founders of G-Force PD, where as a community we try to help each other now moving the field forward in kind of a non-competitive, collaborative manner. Even so now we all started our own companies now that take that work forward. So this have so far grafted 2 patients, and they use a very similar type product, which is also off-the-shelf, large scale. There was 1 patient reported in the past that was basically treated in a way that didn't really require the standard FDA approval because a one-off case, not a real clinical trial. But it's not clear how much that patient really benefited, but it was the first autologous study. Similar to what Aspen wants to do in California, was mentioned before. No, I don't think they grafted any patient yet, but they had clearance. There's another group in South Korea that has, I think, by now dosed 12 patients. But again, they just finished the dosing, so they are now maybe about roughly 2 years behind our study to report on their data. Then there are additional trials planned. I think we heard about Kenai was mentioned before. There's Ole Isaacson at Harvard recently got approval for another autologous trial. So the point I'm trying to make here is that obviously there's quite a high level of saturation already in the field. Not to say there shouldn't be more, but I think it's important that new trials, I think, go a little bit beyond what has been already done. And I think that's going to be now on a much quicker pace some of the other topics I want to touch upon. So what are the challenges of the approach, kind of the approach that I mentioned for ourselves, for example? So I showed you that maybe the higher dose of patient does better, but this is really the best dose. How do you figure it out? It's actually quite challenging. I mentioned also some trials are allogenic, off the shelf, some are autologous, patient by patient. Some groups want to make immune compatible cells, universal cells, so-called hypoimmune cells and so forth. There is questions right now. All the groups put the cells into the target region where the dopamine is missing, but they don't put them exactly at the place where they normally are. Should that be done? It's an interesting question. There are also different stages of cells that are used that might have different potency. And there's also still this issue of graft survival that many cells actually die when you inject them. And that can lead to variability, even if you then just simply add more cells to compensate for that. There's also an importance issue now, if you think about actual translation. I want to do that routinely, hundreds or thousands of patients as a therapy. What is the device you're going to use? You're not always going to have an expert like Vivian, who worked on that for many years in animal models and can load this complicated way the needle. So there's a lot of effort by many to say we need to invest into devices that make that kind of foolproof. And so I think that's an area that's going to be quite important, but that's going to be sponsored by organizations like CERN or done in the industry. We'll have to see. There's also new ways to track patients. Right now we do this UPD rest, time off. These are very crude measures. And so you can imagine there's a lot of information that you can get from wearables, Apple Watches, Fitbits, and so forth. There's a system that actually Blue Rock or Bayer is using, it's called Emerald, where you don't need to even wear a device. You can install it at home, and you can remotely keep track of the movement of patients. And then there's also the question of patient stratification. We know so much more about the disease. The Michael J. Fox Foundation, together with others, invest hundreds of millions to really do this tracking of patients. And so we can now ask, what are the best patients that might benefit from this approach? What are the ones maybe that are less suited? And how can we have maybe biomarkers that help us to study that? What about on the cell side? What's the next generation cells? So there is always, you can make cells always better, no? We could always maybe make them more potent. We think maybe it could make them fully more mature when we inject them. Maybe they need less cell to mature. There would be really no risk at all that the cells could form tumors if they're all post-mitotic. But so these are areas where you could develop, but some people will say, why do you need it? If the other stuff already works. But that's an area that continues to be developed, particularly to get, for example, the right subtypes. This issue of survival, we made actually some major progress. There's a paper coming out soon where we figured out some of the mechanisms why cells die within actually the first few days after grafting. And we have a very simple fix that has to do with TNF-alpha signaling that can basically overcome that. That could be added to such grafts, maybe make them more reliable and requiring less cells. And then finally, that's really a big topic now for the field. can we do cell plus gene? So can we add a gene that helps the cells to be even better than the natural version? So I already mentioned, for example, you could make them such that maybe you don't even need the initial 12 months of immune suppression. Technically, you can do it, that you make cells that are immune evasive, but they have their own risk. The other option is you graft these cells into a patient that has an ongoing disease, and there are ways you might actually be able to protect those cells more effectively. And again, we don't go too much in technical details, but it's the idea that you can lower this disease-causing protein alpha-synuclein that's going to require to transmit the disease to the graft. Well, there's also the idea that you can introduce certain genes and mutations that are actually not only not causing the disease, but might be protective in those neurons. You make up dopamine neurons that might do much, much better in a disease context. Or you could also provide glial cells to do that. So I think these are all interesting areas that could be pursued again for the next generation. There's also a definition now actually that comes there and has to do with what I said before. So if it already works, how do you know it works better? And so sometimes you can just go to larger animal models. And one area, for example, that we want to study is it a good idea to put the cells also back into the location where they normally are in the midbrain? But from there, they need to make very, very long connections back to the target region. So the idea would be you make two sites, one site where the cells are implanted currently, like in the clinical trial, but another site where they're normally located. But we need to have a large animal model to show that this approach really can work over very large distances. The other point is, again, not very sensitive to potency. Some of those assays, when the animal rotates around their own axis, they recover very easily. So if you have now a cell that's 3 times more potent, they're still going to recover the same way. They're already fully recovered. So we need to think more sensitive assays that people try to develop. For example, they use deep learning in the mouse behavior, where they have the animals doing tasks and so forth. So it's going to be very important to look at much more fine behavioral function or functions related to learning and so forth. And these are other animals that maybe mimic the disease a little bit better. This is an animal model from Jim Surmeyer that's very chronic progressive, where first you lose the dopamine fibers where they connect in the brain, and only later the cell bodies. And that's exactly what happens normally in the disease. Unlike the models that we've used for our clinical development, where we just wipe out the dopamine cells in one shot. Or there's another animal that now, Vivian Tabar, I mentioned her name before, we work a lot with her. She uses an animal model that actually has kind of a tremor-like symptom, similar to the patient's, and that has also disease symptoms, this alpha-synuclein in the brain, and see how the cells behave in this disease environment will be very interesting. But again, an area I think that is interesting to actually better understand the interaction of the cells in such a brain. Now that leads me then to the next point, which is what can we do maybe beyond dopamine? So everything that I talked to you about has been to make the dopamine approach better, maybe make it more potent, maybe it doesn't require immune suppression, and so forth. And I think these are all important areas. And again, this is going to be very short because, again, we don't really know too much yet. But I listed, when I gave you the picture now with the patient and with the symptoms, I had some of those points: loss of smell, sleep disorders, gastrointestinal disorder. And if you talk to some of the patients, they can actually be quite debilitating, particularly the GI component. They often really complain quite severely. It's not just a little bit being discomfortable, it can be really quite debilitating. But obviously even more debilitating eventually is the cognitive loss of function. And so again, right now, we don't really have any good approach at this point, and we don't also know exactly everything about cognitive loss in PD. But there's probably two components to it. One has to do more, and where we actually might have a shot to have some impact with the dopamine neurons, is there is some dopaminergic innervation of the forebrain, frontal cortex, that might actually benefit from the new dopamine neurons. And that has to do with kind of what we call executive function that gets worse in patients. So there might be some hope that certain small areas of symptoms might improve cognitively. But the much bigger problem is this diffuse Lewy body disease, but there's no reason to believe that dopamine neuron replacement would do anything major about that and can affect many neuron population. Now, one population that gets affected also quite early, and not just dopamine neurons, they are actually forebrain cholinergic neurons. So they are sitting again in the nucleus in the brain. I think you can see it over here. That basically is an area that projects very, very widely within the brain to all kind of areas, hippocampus, the cortex, and so forth. And they actually die quite early or are affected, dysfunctional, also quite early in Parkinson's disease. And there's a long history actually going back to fetal grafting, similar to dopamine neuron fetal grafting, where people try to have models to kind of have a degeneration of those cells and see where they can be replaced using fetal tissue. And I show you here one such study. That was one of the major ones. It's so-called kind of a cognitive test, a water maze test, where the mouse needs to know where basically a safe exit is on a platform. And the red one is the safe area. And you can see a normal mouse finds this platform quite routinely. A lesion one doesn't. The grafted one is nearly as good as the normal one. And so this is graft of those basal forebrain cholinergic neurons. And we haven't really done much on that work, but I know the Swedish group, Anders Björklund, Agnete Kirkeby, and others, they actually now go back and use some of the protocols we and Sushu Chang developed, refine them further, and actually try, at least in preclinically, see can we have a combined approach? Could you have dopamine neurons for the classic motor disease? And could you have those forebrain cholinergic neurons for some of the cognitive symptoms that you see in AD? Again, clearly wouldn't treat all the symptoms, but that's something which is currently actually being kind of re-pursued within the field. Now, again, there are obviously other ways that can be thought about, and other cell types that people have thought about is particularly cells that would just simply affect the disease process. Maybe a little bit in the cell plus gene, but in this case, it would be probably not the dopamine neurons, but maybe more astrocytes or microglia that could produce antibodies or could be anti-inflammatory to affect the disease process. Now, and even more kind of out there, and again, not something which is going to happen immediately, but technically feasible, is to treat the gastrointestinal symptoms in PD. So we, for example, have shown a couple of years ago that in a very severe model in a mouse, where a mouse actually doesn't have the nerve cells of the gut properly functioning, the same nerve cells that don't properly function in Parkinson's disease because they're affected by the disease, you can literally replace them. So these red spots along the colon are now human cells that take up shop quite quickly in a mouse. And this is now much, much later, something like, I think it was 9 months later, you can see this beautiful fiber. That's a network that corresponds to an enteric nervous system, but it's completely human-derived in a mouse. And what you can show then is that now the gut, the colon actually contracts and releases, as you know, when you push the food forward, which is this blue, orange, red peristaltic movement over time. There's a time axis. You can see that you can trigger these nice waves of peristaltic in a colon completely different with human enteric nervous system. And again, here the idea would be that you would first go to very severe disease like Hirschsprung, show that this can be routinely and safely done. But maybe then it's no longer completely crazy, to use these cells in other disorders, maybe in the context of diabetic gastroparesis, maybe in the context even of Parkinson's disease. So then last but not least, the question is really beyond Parkinson's, and I'm not going to spend more than like 1 minute on that because it's not the topic of today. But I mentioned some of those cells now that we could put in, like glial cells, for example, that could have disease-modifying role. That's clearly the case in Parkinson's disease, but I think where it's already getting pursued is in Alzheimer's disease. This work, as you probably know, can be also supported from Matthew Blurton Jones, but you can actually literally swap microglia. These are human microglia in a mouse brain. We are doing some of those studies too. And you can then have a rejuvenated population because the microglia you have in your brain, they came during development into the brain. They have been there for 50, 60 years, whatever your age is. You can give them easily a cargo, or sometimes we call them, you can make them stealth. You can give them supra-physiological functions that would make them being not only not so much affected by the disease, but help the disease. And so I think these are all very exciting areas, and you can just simply switch the cargo and conditions to then apply that back, for example, to Parkinson's disease, where the mechanisms are slightly different, but microglia and astrocytes play also a very important role, and again, could be similarly swapped, so to say. Another area that's being done there is the same approach that I mentioned with basal forebrain neurons, obviously you could try in AD, has been done before. And I think I mentioned that those interneuron approaches are actually also quite interesting in the context of AD, because there are certain neurophysiological changes that happen that are important for cognition. There's Li Hui Tsai at MIT that actually tries to pursue that, less with interneuron, but by stimulating them, mimicking interneuron electrophysiologically. And then finally, we already heard from Neurona, does really beautiful work with interneurons in California for seizures. I think they're also interested in pain. And there are other types of interneurons that could be interesting for spinal cord injury. So this is just kind of a little bit of an excursion at the very end beyond PD, but I think, again, this goes forth and back to see what are the right cell types that can be pursued by cell therapy. Last but not least, here's some of the conclusions, focusing on Parkinson's. So again, dopamine neuron replacement is now in the clinic, at least in the early stage trial, with later stage trial getting started, promising early results. Many groups are pursuing it, including Big Pharma. So Bayer acquired BlueRock, Novo Nordisk is sponsoring the trials in Europe, and multiple startups. We heard from Kenai and I think Aspen and so forth, in addition to some academic centers. My point is that there could be a big impact is that really try to develop new approaches, not just doing the exact same cells at many more places, but see where really the bottlenecks that I try to highlight. Cell plus gene is one of those approaches. But again, maybe if we want to be very bold, maybe we can also think and invest in the areas to actually target some of the non-dopamine related symptoms in Parkinson's, approaches that could benefit neurodegeneration beyond Parkinson's. And then finally, there's just some of the people. I'm not going to spend much time, but this is some of the crew in my lab that really does the dopamine work currently. But then also, obviously, the whole team needed that's now moved from my lab, at least some of them to BlueRock Therapeutics and lead the efforts over there. I'm going to stop here and obviously be happy to take any of the questions you might have.
Thank you, Lorenz. That's really terrific. Let me launch the questions by asking a fairly simple one. Is the basal forebrain ordinarily receiving dopaminergic input from the substantia nigra, or is its failure an autonomous defect relative to the substantia nigra, or is it just unclear?
I think it's not completely clear, but I would pretty heavily guess that it's unrelated, because there are many other brain regions that are affected. I didn't talk, for example, about the locus coeruleus. That's another population very early affected. It's not a population can be grafted and can be actually generated by Sushu Chang, just published a paper on making locus coeruleus neurons. Very important for some of the sleep-related, mood-related symptoms in PD, but that's even further behind now, trying to develop that. But I think these are all interesting areas.
Yeah, that's quite a lot of overlap with Alzheimer's disease, actually.
Yeah.
Because the locus coeruleus it fails early in AD as well.
Yeah.
Questions from the rest of the group?
JT has his hand raised.
JT, please.
JT, unmute.
Sorry about that. Lorenz, outstanding presentation. Thank you so much. Really appreciate it. Question on IPS work that's used in connection with high throughput screening against neurons in the dish. And I know this is something you were working on a little while ago, but I'm just curious what the latest is. So one of the issues when you obviously reprogram IPS cells into neurons, they're sort of young neurons, if you will, in the dish and haven't fully advanced to maturity where they'd have full manifestation of whatever the phenotypic condition is you're trying to study, whether it's Parkinson's or Alzheimer's or whatever. What's been done to accelerate that process to make drug screening sort of more relevant to later stage disease?
Yeah, I mean, I think there's a lot of early stage developments in those areas. That's an area that we are really interested in. And So we just published 2 papers on what we refer to as improving the maturation state. So there was one paper in Nature, one paper in Nature Biotech earlier this year that has to do with neuronal maturation. So we can add compounds that affect the chromatin state temporarily. So it looks like the way the timing is controlled by a clock. And the human clock basically ticks much slower than the mouse. And it has to do with a certain chromatin state of the cell. And so we can temporarily disrupt that, and then the cells that result after this disruption actually move much faster through the maturation stages. Now, we haven't really used that for either in vivo studies or even for drug discovery. That's actually something we're doing right now, where we try to see, can we integrate that? And so that's one component. It's actually 2 components of maturation. One is removing this, we call it an epigenetic barrier. And the other one is activity related. So you can add factors that control those 2 components to make the neurons go faster. Then we have also a new study that should come out soon. It's not completely out yet. That has to do with a way where we start with Alzheimer's disease, but also in Parkinson's, where we screened every single gene in the human genome that can synergize with the disease susceptibility to trigger disease. They found now a pathway where we can just with a compound, we can stimulate those cells, and they kind of within about 10 days trigger age-like features. Now they're not fully mature, so it's a bit of a different thing between maturation and aging, but they are getting this aging feature that makes them now vulnerable to disease that mimics what happens later in life. So again, those are this area of maturation and age that we now in our lab, and I'm sure other people that do that too, try to integrate to do drug screening. And I think this again an area that's very interesting. I mean, the other thing there is that a lot of efforts are now done to do that not just in one patient. So I mentioned, for example, for patient stratification, this PPMI cohort. We and others have now iPS cells from those hundreds of individuals. And we can grow them not only one by one, but we make pools of them. We call them a village in a dish. You have a whole village of, let's say, 100, 200 patients. Then you give them a drug. You do the aging, whatever you do. And then they should have a disease manifestation. You can now literally see now which of the patients respond to the intervention. In the past, that was very, very difficult because, as you know, growing 100, 200 cells in each well in a synchronized manner is very difficult for some of those cell types. But so this village in a dish, I think, is another recent development. I think that's going to change how people are going to do some of those disease modeling studies. So there's a lot of development, I think, on this area as well for drug discovery that we are very excited. And again, for full disclosure, we started another company which is called Da Capo Brain Science, which is now just getting into the Series A that uses some of those tools together with machine learning tools from the patient data to try to integrate now how do you predict progression, how do you respond to genetic perturbation, how do you respond to drugs. So it's an area I'm very excited about actually, which is always independent of cell therapy, but is actual drug discovery.
Thank you. Very exciting.
Yeah, a lot of great work going on, Lorenz. Other questions from the task force? Okay, so has Jeff Rothstein joined us?
Yes.
Yep, I'm here.
All right. So, Jeff, you've got a tough act to follow here, but I'm sure you'll be more than capable of it. Jeff is an MD, PhD. He's moved around to a lot of places, but he's really spent the bulk of his career at Hopkins, where he has led the Department of Neurosciences for part of his time there. And perhaps most important, he developed the Packard ALS Center, which I had some association with some years ago. And it is a truly powerful discovery engine in the area of ALS. And so Jeff will give us some understanding of what's current in ALS and perhaps where to go next with research. So, Jeff, you're up.
Great, thanks. Thanks, Larry. Nice to spend some time with you. I actually was involved with CERN when it first started many years ago as an external reviewer. I'm going to try to move fast. I know this is a mixed group. As one other bit of background, I'm both a neurologist. I founded the ALS clinic at Hopkins, so I run most, if not all of the ALS clinical trials over the years, but I run a basic science lab working on basic cell biology underlying ALS, of course, with the hopes of finding drugs. What I'm not going to tell you about is cell therapy, And I'm from the East Coast and I'm nothing but blunt. So cell therapy for ALS is a bunch of bullshit. It's a waste of time. The real value in cells in ALS, which is largely a sporadic disease, not unlike Parkinson's. And of course, you just heard the elegant work by Lorenz, and Alzheimer's, is trying to find approaches to sporadic ALS. And there is no animal model of sporadic ALS. And so this is where iPS cells become a real platform. So I'm going to first very quickly tell you about a pathway, but buried in that will be how you can use iPS cells as a platform to understand the pathogenic cascades of the disease, as well as therapies, and then where we're going in the future. So the start of this is very basic, and it's about a new pathway that underlies almost all of sporadic ALS. That real quick background, because I don't know the background of all of you. ALS, like Alzheimer's and Parkinson's, is a largely sporadic disease. This disease largely affects your voluntary motor system. It's due to the loss of cortical motor neurons, which interact with spinal motor neurons and control the contraction of muscles, both voluntary muscles, such as, of course, your arms and your legs, but also your diaphragm and the first part of your swallowing muscles. As such, ALS is a uniformly fatal disease. There are no escapees from this disease. And typically, it progresses over 2 to 3 years, but like all medical diseases, there are exceptions. I've seen well over 10,000 patients, and my fastest patient has been something on the order of 3 months from onset. Others that are 20 and 30 years, so highly variable. Why that's variable is a different issue, but it's always fatal. We used to think of ALS as only a motor system disease. It is not. 50% of patients can develop a mild dementia, and a small percentage, about 10%, overlap with having a frontotemporal dementia, as well as ALS. And we now know some of the genes that cause frontotemporal dementia are the same genes that cause ALS. So although we think of it as a motor system disease, in the early days of the use of stem cells was focusing on motor neurons, that misses the boat. There are many other cell types that degenerate in ALS that contribute to the clinical syndrome of the disease. First gene discovered was SOD1, an antioxidant gene discovered in the early 90s by Bob Brown and Tiku Sadiq. And that led to the first mouse models in any neurodegenerative disease, what's known as the SOD1 mouse. It's used for more than 2 decades. It's a great mouse in that it looks like ALS, and it's a terrible mouse for determining what drugs work in patients. In fact, no model to date has been an exact predictor of what works in patients other than the SOD1 mouse, because turning off that gene, which is a therapy that was just approved by the FDA, shows remarkable efficacy in humans. So know the mutation, you can have a great outcome. And then, with time permits, I'll talk about other mutations. The real change in the disease came with the discovery of a gene called C9orf72, long name, a gene whose protein we don't entirely know what it does, but it's an unusual mutation because it's in the first intron, and that causes a variety of other problems, including aberrant RNA species, as well as aberrant polypeptides that are made from that gene. That gene is actually very common. Up to 20 to 40% of ALS patients that inherited ALS have that gene mutation, and up to 10 to 20% of sporadic patients with no family history have that mutation as well. And it's equally common in frontotemporal dementia. It's the most common disease-causing gene in FTD as well. You can have a family with a sister with FTD with the gene mutation and a brother with ALS. Really a phenomenal focus of research in the last decade or so. Nevertheless, most of the disease is sporadic, and that becomes an important point when we're thinking about how do we understand what's the most effective therapy for the disease. Over the years, using mouse models, and in fact, from about mid-1990s, when the mouse models were first developed heavily here at Hopkins, the community, it's a royal we, use those mice to try to understand pathways, because understanding the pathways, and shown on this slide from a couple of years ago from colleagues in Europe, are the ways you find drugs for the disease. And no matter what you want to study, ultimately it's knowing the disease-causing pathways. And each of these pathways have been described in mice. Now, the unfortunate part of this, and I'm going to come back to this in just a few minutes, is that mouse models are great. They look like the disease, but they may not recapitulate what really happens in human. And quite honestly, the only thing that counts, I'm not interested in curing a mouse or any other organism, a fly or fish. I need to know this is a platform for finding therapies in a human. And the reason I point that out is because over the years, the mouse model, which was distributed worldwide, great tool, was not always matched with human studies to prove that the pathway of discovery, whatever it might be, was also present in humans. And if it's not present in humans, quite frankly, who gives a shit? It only counts when it's present in humans. Many of these have been ultimately described in humans. Actually started with work in my own lab studying astrocytes and excitotoxicity, and that actually led to the first FDA-approved drug for ALS. We've only had about 2 other drugs approved, and none of them work any better than that first drug, unfortunately. And I'm just going to skip through this for the sake of time. What happened was about a decade ago, more than a decade ago, once C9 was discovered, we were fortunate that the NIH sponsored efforts to develop iPS cells for ALS, Huntington's disease, and Parkinson's disease. And I ran the ALS program along with, then I brought in collaborators, Kevin Egan and Chris Henderson, then still in academics, no longer, unfortunately, but they're still great scientists. And from those iPS cells, we began to have a sense of what's really going on in human tissues, even though it's not a complete system. And some diseases, clearly, you can't just use simple 2D iPS cells. But if you're trying to understand the cell biological pathway, you can use that system. And in those days, it took years before a mouse could ever be built around this mutation. And we began to discover that fundamental to some of these mutations, especially ALS, was involvement of the nuclear pore, which I'm going to talk about in a minute. So the nuclear pore, every nucleus has nuclear pores. Think of it as a tennis ball within a tennis ball. And they have about 2,000 to 3,000 nuclear pores. This allows the movement of molecules in and outside the nucleus. Remember, within the nucleus is your DNA, which eventually codes for RNA, which eventually leads to the proteins that are made in every cell. And our body's biology is ultimately about proteins. That entire process of moving in and out is controlled by the nuclear pore. It's one of the largest complexes in the human body. That nuclear pore complex is 1,000 protein molecules assembled in sort of like a donut. And this is actually a schema of what that looks like. This schema of the human nuclear pore structure was actually put together by someone in your state, Andre Holes at Caltech, a collaborator of ours. And he could determine the actual assembly of all of these proteins that make up different domains of this nuclear pore. The nuclear pore not only regulates what moves in and out of the nucleus, RNAs and proteins, but it interacts directly with the DNA as well to regulate gene activation. So really important structure in the cells. And we learned early on, now almost about 8 years ago, that there were defects in the nuclear pore. I'm not going to work you through all of those experiments, but the nuclear pore structure is bound to the nuclear membrane. So think of these individual little donuts on a surface of a tennis ball, has to be bound to the tennis ball. And those are what are known as transmembrane nuclear pore proteins. And we learned that a number of these nuclear pores were missing in sporadic ALS as well as C9 ALS. I'll tell you more about that in just a second. And it all began with the loss of one that led to a cascade, kind of like a domino effect of others. That began with one protein known as POM121. When it's lost from the nuclear pore, there's a series of other nuclear pores that are lost as well. That eventually affects the transport in and out of cells and makes those neurons, especially motor neurons, much more susceptible to death. This is an important cascade because separate from the studies we were doing, others in the field, way back in 2005, had determined that first in ALS, and I'll come back to other diseases in a minute, there's a nuclear protein known as TDP-43. First, we didn't know much about what it does. Again, when I say we, it's the royal we. But we learned that in diseases like ALS and later frontotemporal dementia and even half of Alzheimer's disease, this protein is cleared from the nucleus or partially cleared. It becomes very cytoplasmic, and then eventually it aggregates. And when it's no longer in the nucleus, we've learned that its function to regulate many, hundreds, if not thousands of RNAs are lost. And if you artificially turn off that protein, this was done by another member of your state, Don Cleveland and collaborators, as well as Kevin Egan independently, a whole series of genes are mishandled. In fact, today, individual biotech companies are targeting single genes to replace them. But the reality is all of these genes are mishandled because of this protein not being in the nucleus. And of course, a question would be, why is it not in the nucleus? It's not in the nucleus because of nuclear pore defects. And we discovered that now entirely based, starting from about 2013, using iPS cells. Can't study this in mouse. No mouse develops this defect. This is a common defect of sporadic ALS. No rodent model shows that. But human iPS cells, and I'll get to the question that John asked earlier, and what Lorenz was talking about, maturation, in just a moment. Turns out when you have these cells, and we're looking here at iPS cells that are very young. Sorry, I said that wrong. These are iPS cells turned into early motor neurons, only about a week or two of age. These little green dots are the nuclear pores on the surface of the nucleus. And what happens is there's a protein called CHIMP7, which regulates sort of the structure of nuclear pores, cleans them out, puts them back in. And we learned early on, well before there's any disease in these iPS cells, that this CHIMP7 is relocalized to the nucleus. Shouldn't be there. It should look like controls. You wait a couple of weeks longer, and the nuclear pore may look like the same green dots. It's not. These are nuclear pores, are then missing in ALS. This is 1 of 8 that are missing. And then we wait longer, and then there's a defect in nuclear transport, the function of the nuclear pore. And then we wait even longer, and now that whole TDP-43 process is completely abnormal. So the point of this is, this would be, if this is a mouse experiment, this might be a mouse at 30 days of age, 60 days of age, a young adult, and finally a more senior adult that develops a disease phenotype. So getting back to what you were talking about earlier with Lorenz, we actually do have a pathogenic cascade. I don't care if these are young cells, they're matching, as you'll see in a moment, the pathogenic cascade that occurs in humans. This is not about synaptic biology. In ALS, it's not about synaptic biology. It's more about the cell biology of the cell. And can we see that phenotype? So although people banter around about epigenetic modifications and stuff, that's all bullshit when it comes to this. We see the cascade that's present in patients in the cells as they mature. As you'll see, this is very relevant to our patients as well. Why does this occur is a big question. So let me go back a step and tell you a program that was essential for all of this. If you're looking at a sporadic disease, you can't look at 1 or 2 iPS lines. That's nonsense. That's me in the clinic looking at 1 or 2 patients. I can make no conclusions from looking at 1 or 2 patients. I need hundreds of patients, certainly dozens. And when I designed a program back in 2013 called Answer ALS, the idea there was, let's build iPS lines from not 1 or 2 or 3 patients, because this is a sporadic disease, from 1,000 patients. And Answer ALS was a program that we actually started enrolling in 2016, finished enrolling by about 2021. And it's a big consortium, and it heavily involves actually members of your academic members in your state. We enrolled at 8 clinics around the country. The clinics are shown here, obviously a little heavy on the East Coast. And we really enrolled in clinics that we knew we could enroll fast. We enrolled 1,000 patients a little over a year. Every patient had whole genome sequencing, and every patient, we made iPS cells from their blood. That was done with Clive Svensson at Cedars-Sinai. And in doing 1,000, we uniformly created the same lines with rigorous quality control built into that, reproducibility built into that. And each of those lines were then differentiated to early stage motor neurons, or spinal neurons, not just pure motor neurons. And by the way, that's important. Some people want to look at a single cell type. ALS is not about just motor neurons. I already told you it involves other cells as well as glial cells. We wanted it sort of a sample, the equivalent of a biopsy of the spinal cord. So we did spinal differentiations. And each of those batches of differentiation from each cell then underwent proteome, epigenome, and RNA-seq. In aggregate, along with longitudinal clinical data from every single patient using an iPhone app, IBM Watson was a partner in this. It amounts to about 6 billion data points per patient. We'll get to that later, but what it also meant is that we have an enormous bank of well-differentiated or well-described iPS lines with full clinical data that we could pick among to do our analysis. And the hopes would we find subgroups of this sporadic disease? Because patients, I got to tell you, are all over the place. I told you at the beginning, 3-month survival, 30-year survival. What are these differences? Because in clinical trials, we lump everyone together. And as you'll see later in the talk, we obviously fail at our clinical trials, but understanding the molecular pathways and iPS cells has that potential. In fact, it does show that potential. We can potentially target the right drugs to the right patient. So here's a real world example. I've just told you about these pathways, TP43 and CHIM7, which is an early injury to the nuclear pore. This is a really busy slide, but there's a ton of data. What you're seeing here is something we completed recently. This is 200 patients, individual IPS lines, great reproducibility in producing them, and we wait at different, what it's not showing here, we can look at different time points, 30 days, 45 days, 60 days. And when we do that, we actually see the development of what you're seeing here. This is known as a heat map for those not familiar. And the darker the color, the more dysfunctional that pathway is. You can obviously see these, there are not a lot of changes here because these 30-odd control patients, you and me, if you will, yeah, there's no abnormalities. And by the way, when I say abnormalities, you probably have a hard time seeing that, but this first column is the defect in the pathway that's injuring the nuclear pore complex. All of these C9, FTD, ALS, sporadic ALS, all have almost all of them have abnormalities, or at least 85% in CHIM7. They all have abnormalities in the nuclear pore. And most of the rest of these are those TDP43 products. Remember, I told you there's 1 or 2 or 100 different, if not 1,000. Here we're picking 18 different ones. And very quickly, you can see that, geez, there's a bunch of red here. That's because C9 patients, they have a defect in this pathway that the remaining patients aren't as defective in, even though it's TDP43. Something's different about them. All of the ALS patients in aggregate have TDP43 defects, but it's variable. So right now, for example, biotech companies are focusing on one of them called Stathman. That's this column here, but you can see the red ones of Stathman, there's only about 10% of patients. This would teach us that the IPS platform allows us to tell us maybe which of the patients are better tied to a disease therapy than others. This is really important when we put our trials together, because trials take a long time. It's a fatal disease. You'd like to be assured of positive outcomes. And some patients actually have very little change that are sporadic ALS, which means something's different about them. There's not a nuclear pore. There's not a TDP43 abnormality, even though most people think that everyone has a TDP43 abnormality. So this platform, which, by the way, is showing you the same thing we can see in patients, I'll show you that in a minute, teaches us that for this pathway, you don't need a mouse model. You don't need all that stuff about aging and epigenetics. Not that that can't be important. This pathway is detectable in an IPS platform. Now, does it match, though, human brain? Earlier, I told you it's really important. Whatever model you use matches human brain. Well, fortunately, because of that large Answer ALS program, we actually have autopsies from the same patients that we generated IPS cells from, and we can make the comparison. Did that IPS cell for these pathways teach us the same thing you see at the other end of life? Death. And the answer is, you bet it did. So these are a handful of patients that we've studied, and I'm not going to go through all these. These are the same RNA processing products, but basically the changes that we see in IPS cells, we see in the same patient's brain at death. And that means for this pathway, this model system, and not all model systems this will apply to, is a great reproducer of what's going on in patients. It recapitulates the same changes we can see in patients. And we've even done multiple, it's hard for you to see, but multiple tissue samples from the same patient's cortex, and it's very reproducible. Defects in IPS match defects we see in patients. This is really important if you're going to use a model system. I'm not going to tell you this applies to other pathways that may apply to ALS, but this is a major pathway that underlies sporadic ALS, the vast and common form of ALS. Is this model system going to teach us about inflammation? No, there's no microglia in this pathway, but do I care about that? No, and I'll tell you why in a minute. Can we learn the relationship by using these large numbers of IPS cells to actual clinical indices? Well, we're just beginning to do that. This is where you need AI to come in. And we have collaborators here at Hopkins and as part of the Answer ALS program at MIT to begin to look at that. And some of these pathways in IPS cells do match the clinical indices that we're seeing. In this case, age of onset seems to match the changes we're seeing in the IPS cells that we've looked at so far. Now, by the way, that variability I showed you in IPS cells, we can take a dozen or 2 dozen human brains at autopsy and show actually that same variability. So Stathman or UNC13, these are those single targets that are of great importance right now to some new biotechs. They're changed, but yeah, not every patient has changes. So Again, a recapitulation of what we see in iPS cell can be seen in human brain, teaching us that the real value of iPS cells, stem cells, is actually in human brains. By the way, I haven't told you why it's not so valuable for therapy. Stem cell therapy in ALS has been, quite frankly, a dead end. Terrible companies have been involved, and it's just not the way of getting at the heart of the disease. And part of that is the complexity. It would take, if I could put a motor neuron into someone's spinal cord, it would take a minimum of 3 years to grow that axon to target, even if it could ever find its target. It's not the way to go. It's real. The power of this human platform is in discovering pathways and then using these platforms for drug discovery. In fact, the next slide shows that. So we now know of a pathway. By the way, this is only one of many pathways, but here's a pathway that involved that protein chimp-7 that is involved in the degradation of nuclear pore. We can now take 30 iPS lines that have that defect as shown here, treat them with an antisense oligonucleotide, the fastest therapy to go from laboratory to clinic in neurologic diseases. And we can show in 30 different patients that we completely repair that pathway. This is like a human trial in culture. It's just phenomenal. And this is an example of actually a pathway that's being worked on by, again, another company in your state, Ionis Pharmaceuticals, to develop this ASO for patients. By the way, I stress that that's fast. One of the newer therapies, whether it'll work or not, I don't know, for ALS is one of those TP43 defects known as Stathman-2. Those first papers came out in 2019 by the Cleveland Lab and the Egan Group. And within 3 years, a clinical trial had started using those ASOs. There's nothing that fast. Highly specific targeting agent and incredibly fast moving from lab to the clinic. That's why many of us are quite excited about this approach. There are going to be follow-on approaches, but it's wonderful to understand the molecular pathways, develop a drug targeting the molecular pathway, and bring it to patients. And by the way, do we know this approach can work? SOD1, the first gene discovered, FDA approved the SOD1 antisense. Remember, this is a fatal disease. Certain forms of the SOD1 mutation progress over 9 months. Those patients who've gotten the ASO to SOD1 are alive 2 years later, and they're actually improving in strength. That's unheard of in an adult neurodegenerative disease. And it really is the speed by which we can target a well-defined molecular pathway. It's why the focus on using iPS cells for finding those pathways becomes so important. Why this process begins in ALS then goes back to really trying to understand the defects in the nuclear pore, what initiates this defect. And we've learned that those individual proteins can have, each protein has a molecular code to it, and there can be coding variations. So if this is 1,000 molecules all assembled together, you might imagine that if they don't assemble properly, just because of a small defect, that could activate that degradation pathway. And we have now discovered that, in fact, that can happen. And this is actually a heat map from Andre Holz. The darker the brown, the greater the frequency of defects in those nuclear porins in sporadic ALS can be found. So these aren't mutations, they're called coding variants. And we're beginning to then look for them in those iPS cells. And this coming back to what I showed you, of the iPS cells that we have already, a number of them have these coding variants. We can then use the iPS cells to correct those coding variants using CRISPR methodology to repair them. And when we repair them in patients, will we see an improvement and the loss of the nuclear pore defect and therefore a greater survival? And it'll give us an understanding what was the actual initiating event in sporadic ALS. And this is the kind of analysis that's done. This is that nuclear pore complex. Andre Holz can look at individual proteins and say, well, there's a coding variant here and maybe here, and this predicts that it won't interact with its neighbor protein, and those become candidates for us to attempt to repair. All of this is not possible if not for an iPS-based platform. And I'm going to skip through that. So I mentioned before how important having large numbers of iPS cells are, especially from the sporadic ALS population. This could equally apply to any other disease. A relatively large population of iPS cells have also been developed for frontotemporal dementia. I think the largest one worldwide, though, is really in ALS because of the Answer ALS platform. That data, by the way, everything done in the Answer ALS platform, completely freely available to companies and academics alike. You have to pay a small fee to Clive at Cedars-Sinai to buy the cells only because they have to grow up a new batch. But the data from that platform, all those analytics can be filtered online, completely free. You can download the whole genome sequences. You can download the RNA-seq. You can choose between what ages of patients, how fast they're progressing, whether they have mutations or not, and download that data for free. And in fact, today, almost all 1,000 IPS lines are available, not quite all available. They're generated in batches. From those IPS cells, we roll out new data sets on the epigenomics, the proteomics, the transcriptomics, and of course, whole genome is really known on all of them, all accessible. And to date, the data has been shared through countries really worldwide. This is a little outdated. And projects are started based on those data sets. So we've now already, again, this is a little outdated, more than 95 terabytes of omics data have been released to the world. Hundreds of projects have been started. And as huge numbers of cell lines have been distributed both to pharma and to academics. There are no IP intellectual property restraints on any of this, no ownership. We consented patients from the very beginning, knowing that's exactly how we wanted this data used. And I think that's, one final point though, to almost to end, is that over the years in ALS, this is a list from about a year ago of studies of more than 100 patients in ALS. Just about all of these studies, but one, really is all, have been failures. They're all based on some pathway analysis, many from the ALS mouse, essentially none from the iPS cells. They're relatively new. A high rate of failures is incredibly depressing. However, beginning to look at the genes that cause ALS, either from genetic models or from iPS cells, we're already beginning to show success. SOD1, we already have an FDA approved. The next one, somewhere here, FUS, a relatively rare mutation, is looking almost as good as SOD1 in slowing the disease. In this case, FUS is very young patients with the disease. All of these are, I'm going to argue, easy targets because we know the mutation. It's sporadic disease, which is the vast majority of disease that we have to make a difference in. Now, recently, if we look at what's coming from both iPS cells and some model systems and human tissue, there are a series of pathways that have been discovered more recently that all are potential targets for therapies. And this comes back to where do we want to go in the future? And I would argue where we want to go in the future with iPS. By the way, not all of these are going to be amenable to iPS preparations, especially inflammatory pathways. There you may need organoids. I won't speak to organoids because there's very little work in ALS and organoids. I don't know if they're any better than what we've seen in tissue. I can tell you of all of these lists of pathways, inflammation has been the greatest. If I went back 2 slides earlier to show all those human trials, inflammation is the most common approach to ALS. Every neurodegenerative disease has inflammation. Every neurodegenerative, at least at end stage. Now, 2 weeks prior to the patient's death, do they have that much inflammation? Don't know. We don't tend to look at humans that early. Some companies are beginning to look at biomarkers, but I can tell you all of these inflammatory pathways where often companies have very little data, little histology at death, and that's it. They've all been failures. The greatest list of failures in neurodegeneration are inflammatory pathways. The greatest list of candidate drugs are inflammatory pathways because most of these companies are already dealing with inflammation from peripheral targets, peripheral inflammation. So they have a drug already and they'll come to us. Even though there's a high failure rate, I'm not going to turn a company down as long as it's done, the design is done well, because I can't tell you I can predict what works in ALS, but I can tell you there's a long history, 2+ decades of inflammatory mediators all being failures. The successes are coming from understanding pathways. Obviously, gene targets are pathways. There are now targets focusing on astrocytes, oligodendroglia. Some are not as well worked out. Many genetic subtypes are going to be targets. That makes perfect sense. And what we're learning from iPS cells, RNA binding protein, nuclear pores, nuclear transport, are sort of the upcoming pathways. DNA damage is of interest, and certainly axon regrowth. There are companies that are developing drugs for axon regrowth. Again, great pathways for studying in iPS cells due to growth of axons. These are not platforms particularly good for multiple synaptic interactions, but ALS is not generally thought to be a multisynaptic interaction disease. It's heavily based on true cell biology of individual cells, other than cell-cell interactions like glial cells interacting with neurons. That may be where iPS cells fall short. Organoids might be better. So this is sort of just a current list of what we as a community think are interesting pathways. Most of these, though, can be optimized through the use of patient IPS lines. And I would argue, depending on as you look forward, I wouldn't waste, I'm going to be harsh here, I would not waste your money on cell therapies. And I can answer questions about that. This is what you use IPS cells for finding pathways. And the question earlier about drug screening, no question, in CTRU in San Francisco is already using large numbers of the Answer ALS IPS lines, ultimately for drug pathway and drug screening. There's no question that's an exciting platform for companies, and to a lesser extent, academics to use. I went kind of fast, but I wanted to make sure we had time for questions. I should point out that, like Lorenz, much of what we do in the field is heavily based on collaborative interactions between my lab, Dr. Coyne's lab here for some of the nuclear pore biology, but many researchers, both West, East Coast, and in Europe, that help make all of this research possible. And all of us, I believe, rely on many different sources of our funding to make sure we can move forward rapidly. Thanks.
Jeff, that was absolutely terrific. So a couple of comments and then a question. The comment is, you and Clyde and collaborators deserve a great thank you for not only creating this library, but making it very straightforward for the rest of the community to access it. It's a really powerful discovery tool, I think. My second comment is actually a question. I mean, this may be loony, but the defects in the nuclear pore in some ways are reminiscent of the defects in progeria.
Yes.
And I guess the question is, would all of these nuclear pore defects in ALS benefit from a shot of the normal progerin gene?
So different. So progeria, I believe, affects lamins. I can't remember if it's lamin A or I never remember this. Lamin A.
Yeah, fair enough. Right.
Nuclear membranes are actually not the defect in ALS. Believe me, we've looked at it. We do a structural illumination microscopy. We'd love to do cryo-EM. It's very difficult in iPS cells, but we've extensively looked. So nuclear lamins are not really defective in ALS. It's the pore itself. But it was a very important question for us to look at earlier. On the other hand, could I tell you that if we didn't somehow enhance lamins, or for that matter, this maybe go beyond many of you in the audience, the link complex, there are certain link complex proteins that interact with the nuclear pore, such as SUN1. There may be a hint there. Right now, most of it's coming from this escort3 pathway, which is CHIM7, and its interaction. We don't know what starts that. We guess right now it's these coding variants that we think are somehow over time build up of some slowly developing injury. One of the things, again, for any of the scientists in the room, and again, this is where it comes from the Salk, is that nuclear pore proteins are some of the longest lived proteins in the human body. Some of those nuclear porins have half-life measured in years, not hours, not days, but years. And in fact, when we first published our first studies in 2015 on nuclear transport defects, it was Rusty Gage who separately that same year had a paper in Science on defects associated with aging. And one of the highest one was a nuclear porin. I can't remember if it was NUP62 or Group 98. So aging, yes, in some way is that component, but I can only hand wave at this point, Larry.
Interesting. Questions from the group? I mean,
by the way, go right ahead. Someone has questions.
Oh, well, I was just going to mention, on the one hand, it's a tremendously disappointing therapy discovery effort so far. I've certainly seen those firsthand. On the other hand, I think where you are with the nuclear pore is a really incredible opportunity to make a difference for a subset of ALS patients. If you can find ASOs or drugs that help.
Yeah, I want to make sure I did this fast, that there are other laboratories that are very much IPS based. A lot of laboratories in ALS are beginning to move away from mice using IPS. But and you heard me being very blunt at the beginning, it's not about therapy. It's what I mean by that. It's not about cellular therapy. It's really about using the value of a true human model system to understand disease, as long as you make sure you try to replicate that in human brain, knowing you might not. That's end stage disease. A lot of other things happen at the end stage, but not cellular therapy. And I know my colleague Clive has been trying cell therapy, but it's the complexity in a disease like ALS. This is very different than Lorenz's discussions around, say, Parkinson's disease, which is very focal. You're talking about the entire motor cortex, the entire neuroaxis, your entire spinal cord motor neurons, which have very long processes. The soma, the cell body is, as just to those of you who don't know, the cell body of a motor neuron that allows you to wiggle your big toe starts at your belly button. And that long axon goes from there all the way down 3 or 4 feet, depending how tall or short you are. And that's just incredibly complex. Not to say one day someone might know how to do that, but those days of focusing on that, I think, are past. And the few companies in our field that have begun to deal with cellular-based therapies have been terrible. I can only say, I can't say anything nice about them. They're not good companies. John, you have your hand up.
JT, yeah.
Yeah, hi, Jeff, and another just outstanding presentation. Thank you. If you had to speculate, what is it that allows the patients with the condition for 25 to 30 years to make it that far, let alone Stephen Hawking, who Clive hosted at Cedars a number of years ago, which was a fascinating event. Why is that happening?
Yeah. So you're asking it's a great question and the simple answer is shit, I don't know. No one knows. All degenerative diseases are late onset. Alzheimer's, Parkinson's, you could pose that question to any one of us, and we don't know that answer. We know this is where animal models are useful, or even the IPS. We know they can have an injury cascade early on, but what tips that over to cell death? We don't know yet. Is that an environmental stressor? We know the IPS clearly has a defect. I can show it. I can show the same defects in brain, so I know I can bridge those two. Even mouse models, we knew you can see a defect. Mouse may die at 120 days of age, but gee, at 2 weeks of age, we can find already early RNA changes, some protein changes. Why that accumulates to reach a threshold, I don't think any of us really fully understand. I can hand wave, but say, well, nuclear proteins have such a long half-life, and they're not dividing cells, so it can take time to build up an injury. I'm really hand waving there. So that was a really crappy answer to your question, but it's the best I can do. Stephen Hawking, though, people don't realize he was on a ventilator. If you're on a ventilator, you don't die of ALS. You die because of ventilator, you get a bad infection. I've had patients who lived 20 plus to 30 years when they should have died when they were 10 years into disease, but they are on a ventilator. The disease does not stop. And you could see Stephen Hawking couldn't do much. In fact, in the beginning, as the disease moves on, the only thing preserved are your eye movements, and we use computer screens to read letters, but eventually even that can go away for motor neurons. But as long as you're breathing, you don't die. And you'll only die because the ventilator comes off accidentally. It has in patients occasionally. Power to the ventilator, or you develop a severe infection. And the fourth reason is you finally say, I don't want to be on a ventilator anymore. Help me end my life. And all of those are real world examples. But he lived longer because he was artificially ventilated.
Interesting. Vito, you have a question.
Hi. Yeah, I have a question for Jeffrey. Jeffrey, I got to tell you that my PhD from Cornell is in the humanities. Okay. So, but a question, I read in the newspapers that polio is resurgent in New York City's waters. And I'm thinking, is there any interest in your community to understand how the anterior horn cells that are killed off by the polio virus and then the surviving neurons, I presume, arborize to pick up orphan neuromuscular? Is there anything in that area? I don't read anything in the post-polio.
So, yeah, so what you're referring to is what's called post-polio. First of all, it's extremely rare. I have, I think, 3 patients in my career that are post-polio. One was a senator. There was actually, the only researcher I know in the US was a guy named Bert Jubel at Syracuse, who used to do work on that. Beyond that, I know no one else doing it. There's a version of that. Some of the childhood encephalitides, both West Nile and I'm blocking on the other one. There's actually a colleague who was a young MD, PhD, pediatric neurologist was working on one of those where it looks like polio. It's a resurgence of a virus like polio. It's not polio, affected children predominantly, motor neuron specificity. There it had to do more with the virus getting into motor neurons. And by the way, affecting the nucleus, including nuclear pore proteins. And that's actually what he was studying in my lab. But the post-polio side of things, I don't know of anyone studying it. It's exactly as you described. It's the arborization. And actually, as you said that, it reminds me, as we're keeping SOD1 patients alive now who've lost half their motor neurons, we're seeing what we think is that same effect. The remaining motor neurons branch out now.
Yeah.
And for those on the audience who don't know this, post-polio is the fact that if you have polio and you're very weak, your other motor neurons have sort of compensated. They're helping out, keeping things moving a little bit, but eventually they die as well. And that was called post-polio. Eventually you're stable and then you start getting weak again. The same could occur with the SOD1 patients, except they're getting their therapies today much later in life, their 50s. But I have to tell you one other thing that's the most exciting to many of us. As we develop gene therapies, it's not as much about stem cells, but it derives from them. As we develop gene therapies for patients who have disease now, what we're doing is thinking about the next step. So what if you have ALS, unfortunately, but so does your son carry the mutation? Well, he's going to get disease one day. Some of these are 100% penetrant. We're actually following those individuals now. And when there's a protein in the blood that says the nervous system is starting to get affected, they're getting the gene therapy before they get disease. But the idea is they will never actually get ALS because we're turning off that gene with this therapy. And the next generation of that, and this is going on heavily actually in some of the people in your state, someone I'm trying to recruit to Hopkins, is using CRISPR to completely fix the gene. But right now, ASOs are that next great hope. And that's preventative neurologic medicine, unheard of in neurology until therapies. And I'm going to argue this is where IPS platform, you're going to invest locally in your state in that. Those are the things to think about, that forward view.
Thank you.
So if there's nothing burning, we should probably wrap up this discussion. Jeff, fascinating as always. We do have a couple minutes for public comment. Claudette, do we have any of that?
We actually do have a public comment from a person in our room.
Hello, my name is Stuart Chambers. If that rings a bell, I was a postdoc in Lorenz Studer's lab. I developed many of the protocols that were for making neurons in the nervous system from pluripotent stem cells. And that's, the basis for what now propels Blue Rock, Neurona, and Aspen Neuroscience, as many others, towards the clinic. I'm here to advocate for early seed startups and their application process as part of applying to CERN. I wanted to bring this to the task force's attention because there's a big challenge with pre-seed companies in particular in regards to applying for CERN funding. There's a solvency check in place that makes it tricky, basically makes it a burden. In my personal case, I could not apply at all. And I just simply wanted to say that, that this is important. It's important that we figure out ways for pre-seed and early seed companies to continue to be able to apply for CERN. I recognize the purpose of the solvency check in terms of making good investments and being good stewards of the California state's money. But at the same time, I wonder if there are creative solutions to this problem. And I just simply want to bring it up to this audience and CERN as a whole so that we can talk about this in the future.
Great point. Can somebody make sure that Shyam Patel gets on this to think about it?
Will do, Larry.
Great. Any other public comment?
And no hands raised.
Okay. If not, I'm going to adjourn us 2 minutes late. Sorry for the extra time. And thank you all for your participation. Fascinating discussions today. Jeff and Lorenz, thank you for helping us. Vito's got it right.